This is a production of Cornell University.
All right, thanks, Jalu, for that introduction, and thank you everyone for coming around. It's been a long road, but at least we're going to have a headway today. So I'm presenting on high-throughput phenotyping and then genomics-assisted breeding for quality traits in cassava. And maybe for the record, my full name is Ugochukwu Ikeogu. I saw the advert, Ugo. It has landed me into trouble in China. So they're like, we're seeing Ugo and your name is Ugochukwu. So, well, it's okay. So I hope, Geneva, I don't know if anybody is from there, is connected there. Yeah, I guess this should be okay. Yeah. So I'll be giving an account of my 5-year stay in Cornell, and then I will talk about a little bit background on cassava, 3 studies that are part of my dissertation, and then some other things, like John Luke said, we're a little bit trying to see things change in cassava. So there are a few other studies that are not reported, but I'll just mention them. Well, the first thing, looking forward and then thinking globally, we need to avoid global nutrition crisis, which should be the vision of everyone here as a scientist. One thing to note here or see is like there are millions of people who are in need of food all around us. And another thing I want to mention is that this is from USAID, that food assistance needs, that of 2017, it was unprecedented. So while we have some basic drought-related things we can predict, okay, we might need so amount of food, there are a lot of natural hazards and all the rest of them, insecurity problems that can lead to food insecurity and sudden need for food around us. So I like this picture. What do we need to do? We need to be proactive. So if we don't prepare, we have a little space and then we have a lot of need. People are hungry, and then if we don't have enough room, create or produce more food, a time will come, maybe a lot of people will be hanging around, and then that will be disastrous. So a little bit about cassava. When we are talking about global food crisis, cassava is one of those crops we can depend on to help us manage global food crisis. I got this quote from FAO, and it said that cassava was once regarded as the food of the poor, but is now becoming one of the crops that nations depend upon, and then a crop that is affecting the global economy of a lot of nations and part of the developing countries, sub-Saharan Africa. Cassava is a key crop that we look up to. It's so important because cassava, more than 800 million people depend on cassava daily and globally. And it's a unique crop too. It's hardy and low input crop. Cassava can thrive well in marginal areas. And one other thing I want to emphasize upon is that even though cassava is a food crop, it's also one of the crops to depend upon to industrialize agriculture in most of the developing countries. This is another quote I saw, and then trying to compare it to the present condition where we are. So despite its potentials, cassava has the lowest research intensities. So it has been like an abandoned crop for a while, but I want to say that some of those are changing. We are in a period I tag as a new era in cassava research. And that era, I think, is marked with a lot of increase in funding. NextGen is one of it, and basics and all that. I think Gates Foundation alone, I don't know how much, I don't have the figure here. There are a lot of investment and interest in cassava research currently. We have abundance of genomic information, improvement in computational and analytical techniques. There's upsurge in human and material capacity, and currently there is a heightening demand for cassava for food and biofuel. especially in Asia and Europe. So the ongoing developments are creating the urgent need for us to reshape how we do things and then meet up with the rising demand for cassava products. But in doing so, we need to understand cassava breeding challenges. Our ultimate goal is to increase genetic gain over time, and then that we can do when we take care of so many important factors, increase our selection accuracy, selection intensity, and above all, if we can reduce the breeding cycle of cassava, which by the way has a very lengthy breeding cycle. It takes up to a decade for you to release a variety. And cassava has a poor flowering and seed set. We have limited germplasm and diversity, and we also have the need to get exposed or have access to better tools, and that could range from phenotyping and data management tools. So maybe a little bit distraction. So that leads to some of the things I'll be discussing. One is the first thing that is part of my dissertation is to look at the problem of phenotyping in cassava. So I titled this, the first study, rapid quantification of quality traits in cassava using portable near-infrared spectrometer. Well, I need to mention here that most of the quality traits we want to improve on cassava. We don't have a lot of high throughput and standardized method of phenotyping them. And I want to give an example using these two traits. These are carotenoid evaluation. Usually it's easy to separate between yellow and white clones, but when you have a lot of variation in yellowness, it becomes difficult. And what we usually do is a visual assessment. We can go far to go into laboratory extraction or using high performance liquid HPLC chromatography. But one thing with all these methods is that one, they need laboratory, you need standard laboratory equipment, you need skilled technicians. And when we are talking about low resource breeding institutions, some of these things could be challenging, having constant light and power and all that. So we started thinking about, and these are indirect methods. They say visual scoring is an indirect method of quantifying carotenoid. And instead of just getting a feel of the total carotenoid, we are thinking about carotenoids have some other individual components. So we are just at the point of quantifying for total carotenoids. And then going over to dry matter, which cassava on its own is almost 60 to 70% water. And then for whatever valuable component that will come out from cassava, we need to have a good feel of the dry matter evaluation. So we use the oven method and specific gravity method. Also, like what I emphasize here, is still time consuming and low precision. Specific gravity is just what you're seeing in the diagram here. You weigh it in air, dip it in water, try to get the specific gravity, and then from there derive dry matter content and starch. Well, people have been using it, but currently there is a development in the use of NIRS technology, and I just want to show like electromagnetic radiation scale. When we talk about NIRS, we have visible light here covering from 400 to 700 nanometer, and NIRS per se is between 700 to 1100 nanometer, and then you can have mid-infrared that covers up to 2500. So a little bit about NIRS. NIRS studies the interaction between electromagnetic radiation and then matter. So earlier studies have proved that once you expose a matter to electromagnetic energy, it produces frequencies or vibrational frequencies that you can use to quantify the chemical component of that matter. So they have used it over time to quantify and then quantify so many known molecular components in many food samples. They are using it in health and in so many other places. What are the advantages of using NIRS in phenotyping? I need to mention here that the NIRS technology has been evolving. We used to have a tabletop. It's still available, but the recent developments, we are having handheld, and currently, maybe you see some people have my GWAS lab is working on this currently. You are having NIRS tools in a very portable format. And one maybe good thing to think about, looking at the ranges and all the rest of them. So like I explained in the previous slides, looking at what you want to measure, whether it's just NIRS, mid-NIRS, infrared, or covering between visible and near infrared. So most of the things I did was with the handheld, which covers both the visible and mid-near infrared region. So NIRS is fast. We can scan a sample in a matter of seconds. It's non-destructive and inexpensive. It's versatile, and then one thing to know is that from the same one spectrum, you can, if you have a good calibration, you can derive a lot of traits at the same time. It's here when we are using NIRS, we are not talking about waste and then other hazard-related disposal and that are associated with use of chemical and laboratory method. We can perform, anybody can, once you train people, this is just like shooting a gun. You just shoot and then it takes the spectrum. So you don't need to be a scientist, a chemist to use NIRS to collect data. So a little bit about application of NIRS. You don't just get it and then you have to do a little work about before you start using NIRS in your phenotyping. So we go through calibration phase, and what it involves is that first you want to match your spectral data with a reference method. So for whatever trait you're interested in, I maybe talk more about carotenoids. If we're interested in total carotenoid, we want to match the spectral value we have, and then you have to go through a little bit of spectral pretreatment to correct for scattering and then differences in nature of sample and all that, and then match it with a reference method value you get from the lab or whatever that looks like the standard for measuring that trait. Then you have to train your model, develop a calibration model, train, and then have your validation before you can use it to process samples that have spectral value but don't have laboratory values. So for us in Cassava, one of the things we are trying out is to have a field base, because part of the things we consider before going for this is we don't just want to get a desktop that it sits in the lab, and also the one thing I didn't mention is that the desktop is also can be affected by temperature and all that. So you also need light source and all the rest of them. So it's almost like having a standard laboratory too. So we needed something that is field-based, and then that's why we went for this. And we are asking, can we just, here we are cutting a cassava root and then scanning from that region and then take some scans and then use it in the lab instead of coming back to the lab or carrying things. Here's a picture of one of our outstations. So some of them are about 8 hours drive from the normal station. So you don't want to start establishing laboratories in outstations and all that. So one is that we are trying to develop calibration on intact cassava root. And the next thing we're doing, also trying to just create things. So we went into construction, get somebody that can help us with a grinder that doesn't depend on electricity, so that you can carry this all around, and then even field-based to grind and then homogenize, because for some traits in cassava, maybe I'll show you a picture of, if you have seen cassava, you have a variation in trait concentration and some trait concentration along the root. So we want to homogenize and then see how that can also improve our calibration accuracy. So for objective of this study is to develop calibration for major traits, carotenoids. When we talk about carotenoids, we have been presenting most time total carotenoids, but let's just remember that we have a lot of components of carotenoids that are also that have health benefits. Example is lutein. While we talk about night blindness, trying to correct for carotenoid, lutein and other traits are actually active, though they are not provitamin A carotenoids, but they are important in taking care of eye-related problems because they absorb the ultraviolet rays that protects your eye against cataracts and all the rest of them. So let's also not just focus on total carotenoids. So our aim is to develop calibration as much as we can for individual carotenoids. Dry matter content, and then, like I said earlier on, develop and probably compare calibrations between intact and matched roots, and then we try to see how far we've gone comparing specific gravity method and oven method from using our NIRS-derived dry matter content to compare with the existing method for dry matter. So for reference and calibration development, throughout my work, I had a lot of collaborations. We developed calibration with people in Seattle, Colombia. Some were developed in National Rural Research Institute, Umudike. And for carotenoids, we use HPLC as a reference and lab extraction method in NRCRI. For dry matter, mostly we use oven drying and then specific gravity method. For models, I compared a lot of pretreatment. I mentioned earlier that you don't just, your spectrum has a lot of noise in addition to the real information you are interested in. So you have to see how much you can remove the noise so that you can focus on the real thing. We tried both linear and nonlinear models, and then for validation, tried cross-validation between training and validation sets, divided the calibration set we had into two. So I'm showing some results to just emphasize that when we're comparing calibration between intact and matched roots, I'm showing result on total carotenoids, all trans beta carotenoid, and then total carotenoid at 2 mudike, just to prove like from here, I mean, there are a lot of statistics to use to check your calibration model, but one of them is arrow square in cross-validation. And having this high value gives us the assurance that we are doing something even from intact root, even though when you compare in most of the other traits like dry matter content, we had better calibration, we matched than the intact, but they are still high enough that we can use for screening in the field. Another thing we try doing is to compare between the methods we're using. So here, try developing calibration using specific gravity as a reference, and then using oven method also as a reference. So that gives us like the real value for specific gravity. And then when you fit a model with specific gravity, you have a NIRS, NIRS value, predicted value from intact root using specific gravity as a reference. The same thing for using the matched cassava root samples, then using the oven, and you have a prediction from NIRS, from the intact and matched. What we can see here is that, well, specific gravity, one thing we also did was to try to make sure we remove outliers. I will emphasize that we had a lot of outliers in specific gravity, and if you just use the raw data, the correlation was really low. But we tried looking at it and then removing things that we felt like were really high. But with that too, we still had like the relationship between oven method and specific gravity method is around 0.64. And one thing to report as concerning use of NIRS is that if we develop calibration with the oven method and then comparing it with NIRS predicted, we're having high correlations that gives us that confidence that whatever that is predicted from the NIRS is something we can use that is close to the values we get if we're drying it in the oven. So I tried looking at some laboratory values to check what's the cost, are we saving anything in deploying NIRS for phenotyping. I looked at lab, you can ask me for the names, I removed them for maybe security purposes. So I looked at 2 laboratories that are doing NIRS. One, so these are different laboratories. So if you want to use the starch enzymatic or calorimetric method in quantifying for starch, that takes you up to like $75 per a sample. And I estimated that for us in a breeding program like NIRS, we have multi-locations. On average, we are certain to measure up to have up to 4,000 samples in a year. And if you calculate that, that's giving you more than $300,000. But if we are using NIRS, this is another laboratory. They are just specialized in using NIRS for calibrating for or phenotyping for starch. That value drops to 19. And then for us now, going back to maybe a national breeding program, if I didn't use up to 200, but if I estimate that both calibration and then validating my model every year, I'm spending up to or investing up to such amount in building a reference. All I will be doing is having spending is about 0.05 of this amount. So in other words, if we apply this, we are sure that we are saving a lot for the breeding programs. And this is not just, I'm using one trait, we are quantifying for carotenoids. This is not total carotenoids. If you go for selective, you have up to, if you select more that like what I've seen in HPLC with individual carotenoids, you're having up to $125 per sample. So with this, I think I want to conclude that NIRS provide a good phenotyping alternative for the evaluation and improvement of many traits. And then this can also help us discover some novel traits that we have not been quantifying in cassava breeding. The portable NIRS is highly adaptable for field-based screening. And then even though we had higher calibration in MASH than on intact roots, what we had and calibration results we have from the intact root is good enough for screening purposes. And then this saves time and then production cost. The next study is to go back, have a little more detailed study on carotenoids. And since NIRS opened the door for us to measure individual carotenoids, so we want to see how the individual carotenoids correlate with total carotenoid. So this is one of the ambassadors of cassava. It's, it's easy here for you to, a little background. This is a yellow cassava. I don't know how it's looking. And these yellow ones, and if you see far away here, the difference is clear. But then in terms of quantity, we don't have so much to say about it. And not just that we want to check how much we have in the raw samples, it will be interested to also know the retention of carotenoids when we have the products as Gary and others. So one thing to note is that most of the studies will focus on, and I've laid emphasis on this, on total carotenoid, but it would be good to go deeper into the individual carotenoids. So the objectives here will be to understand both the phenotypic and genotypic correlation between, especially between TCC and other individual ones, to know why we're improving total carotenoid, whether we're also improving the other individual carotenoids. If we can understand some of the studies that focus on TCC, do we know the regions that are associated with variation and those individual carotenoids, and then check the option of using GS in improving carotenoids content in cassava. So one thing we used here is that we had about 994 clones, and we analyzed this using a non-linear. Results I showed were mainly linear model from partial least square regression. We tried a non-linear model because one, apart from non-linear models capturing non-linear relationship between your predictive and dependent and independent variables, non-random helps you to capture other relationships that are not there. And then also is a little bit direct and then makes your results more interpretable than linear models that are based on latent or principal component factors. So we try using nonlinear model to analyze this, and then Umudike has like 2 trials. The training population is just based on when they were collected, and then they have been managed in 2 like replications, but we just call them TP1 and TP2. We had 2 replications per clone in a trial, and then for NIRS, we had 2 to 3 samples per plot. Location, we have data from Umudike, Tobi, and Kanu, and we use more than 100,000 SNP markers for this, and then we focus on these traits that I've also mentioned before. So the result I'll be showing here, most of them are closely related to genotypic and phenotypic correlation. So here I'm showing the narrow sense heritability for all the traits. And then for the correlations, we saw, more importantly, one of the encouraging things is that is also shown before that all trans beta carotene, which gives you a value of beta carotene content, is highly correlated with total carotenoids. So we are so sure that while we're improving total carotenoids, we're having a good feel of all trans beta carotene, which is like total beta carotene. There are other isomers of beta carotene, which are also, they have also high correlation with TCC. But one good thing to report here is that for all the traits we studied, they have positive correlation with TCC, which is encouraging. We saw some levels of negative correlation between the traits, but I think it's a good news to know that while we are striving to improve total carotenoid, we're improving the other individual carotenoids too. For GS model, I tried to fit in some models to check prediction for carotenoids. So I will show this. I hope you remember it, but maybe I will emphasize on it the next slide so that you don't forget. So one thing is that usually when you have your raw data, is like a usual practice, we do a two-stage approach. First, fit in your model and then take care of different factors and get your blob, use your blob like estimated value to fit in the genomic blob so that you can get your estimated breeding values. But the one stage, this is like a prosy to the one stage, means fitting in everything from different locations at the same time. So, and then we report calibration using single trait and multi-trait. We have seen that most times you can improve the prediction of low heritable traits using it that are highly correlated with other traits. And we tried using multi-trait method also. And then we compared that with nonlinear methods using random forest too. Don't worry, I think I tried to cover it so that you can remember. So you won't have trouble trying to remember what was the prediction accuracy for this. So once more, don't forget that this is single trait, two-stage prediction accuracy. This is a multi-trait linear G-blob linear model using single multi-trait. And here you have the random forest in two-stage. Here you have single trait using one stage and multi-trait one stage, and then nonlinear random forest one stage. So we have values for that trait, but I'm trying to use these three to illustrate that most times what we saw is that multi-trait to some extent we're doing a little bit better on average compared to the single trait model, but that reversed when we use the single stage model. So, and then overall, using nonlinear model was doing a little bit better than the linear model. So the result is about the same for other traits. The next thing we did was to fit a GWAS model for all the traits. I hope my stars are really showing very well. I tried expanding and realized they scattered and moved to other columns. So one thing we found out is that even though we didn't have any significant hit on alpha carotenoids and phytotin, for other traits, we were able to discover some regions that were significant for most of the traits we studied, especially in chromosome 1. For lutein, we have up to about 5 regions. And then for total carotenoids and most of the beta-carotene components, they were almost showing significant at chromosome 13. So overall, we were able to look at the genes that are related to carotenoid in cassava. And we saw like 2 basic genes that have been reported. One, this has been reported for TCC, and now we are reporting it not just for TCC, but for other individual traits. And then this also also another gene we found out within those regions. But then for the other significant regions, we didn't see a lot. This has been reported in many studies. We didn't see, but we found a lot of genes that have biological functions that we believe that maybe further studies can help us understand their relationship with carotenoids and all that. So in conclusion, we have seen that if we're utilizing NIRS, it will facilitate not just understanding carotenoid, it will change the way we are doing things in phenotyping, and then that gives us a hope in further studying the relationship between TCC and other component traits that we have not been looking at, and that can help us redefine our selection index and all that. One good news is that we got a positive correlation between TCC and the individual traits, and we verified a major trait region that was been reported for variation in TCC alongside other regions that have not been previously reported. And we have shown with the level of accuracies we got that we can use GS to improve carotenoids in cassava. So quickly, trying to make sure I beat time. The last study that is part of my dissertation is part of the things I said. We have access to SNP marker information, and we're asking how can we use it to redefine nursery system in cassava. So a little bit background, I'm showing a polycross we have at Ubiaja. And one thing we need to look back at is that in making control crosses in cassava is very difficult. One is a little bit obvious. We have clones that flower early, 2 months, from 2 months, 3 months after planting, and other ones that delay. But within the same clones that flower within the same flowering, that have the same flowering habit, we see a lot of variable flowering habit. Some clones they, open up early in the day. So if you are used to crosses, maybe it may be difficult. But this is something I did like every day. So some clones will open between early in the hours of the morning, while some clones you have a crossing scheme and you're waiting for the female, maybe the male has opened, and then where you need to collect flower, the other one has not opened. You have to wait some time before you go to the next plot and come back, you realize that it has opened behind you. So there are a lot of variation and then a lot of magic in cassava that you need to love before you can endure and work in cassava. The cost also, so one of the things I did, like John Luke said, is managing casual workers. We hire, sometimes you have to hire up to 20 people for crosses and all the rest of them. It's not cheap at all. And producing botanical seed, when you are doing had all those kind of variation, and then to get seeds that you can completely say that these are true crosses, most times using up to designing a scheme of up to 20 clones and all the rest in a nursery scheme, you might end up getting 5,000 in a year. And that is not good enough for any valuable breeding program. So that is part of the things we are looking at. And what had previously breeders have tried relying on form of open pollination to improve increase the production of botanical seeds. But one problem, and one of them is polycross, which is more like isolation, and then you pick your clones that you're interested in and then plant them in isolation and allow them to mix naturally. So you don't have the problem of trying to control it. But the problem here is that you lack food parental information and such. You don't know the pollen donor and all the rest. So they have used it, and then we're asking the question, can we use the available marker we have to see if we can resolve and modify this kind of scheme in cassava? So you can get a lot of seeds from open pollination, but then you need to match each of them, the progenies, to their true parents. So what we did was to design a polycross using the same method. I'm just showing a kind of design on what you have to do. You need a good design so that you can implement this. It's not like, well, cassava is naturally an outcrossing crop, but you still need to introduce a lot of design. So let's see, this is one clone. So you want to make sure you randomize it such that you have the possibility, increase the probability of each cross intermating with a neighboring clone that is not itself. So for this, we selected 29 clones from our genomic selection pipeline, those that have high GBVs. And one thing also we considered was trying to be sure that those clones have about the same flowering habit so that we don't introduce. Well, cassava is an issue, but then you still see that between some flowers, though they flower early, but they still have an overlap between the long ones. So, but we try to choose the ones that have an average, the similar flowering habit. We try bringing randomization using modified this quasi Latin square design. And then for isolation, we tried observing 50 meters, looking for a field that has a kind of 50 meters distance from any neighboring cassava. For Obiaja, we succeeded, we were lucky. We spoke with the yam breeding program, so they gave us a site within their yam breeding program and yam experiment. So we had a lot of yam around this side, and then we were sure that we don't have any neighboring cassava clone around. But for this study, well, the effect of the seed production, we succeeded in getting up to 17,000 seeds that we processed. And for Ubiaja, there were more than 25,000 people just harvested. And I know that Ubiaja produces more, supports more flowering and seed production than Umudike. But that shows us that with this, in terms of seed production, at least, we are covered, and then we are faced with the issue of is it possible we can at least derive or have a hold on the parental or clones that contributed to those progenies. So we use about 3 to 8 seeds per clone out of the 29 replications we had in the field. And one approach of the parentage is using relationship metrics. Our idea is that if genomic markers across the genome can be used to measure genetic similarities, can we look at the genomic metrics and then use the coefficients of relationship metrics to say something about parentage and relationship of clones, progenies with their parents. So for that, also, we tried another approach using penalized regression. So we looked at this as a regression problem and trying to fit the genotype of each progeny at a time against that of all the putative parents that are involved in the scheme, and then get the coefficients, and then using lasso as a restriction to pull down some coefficients that are around 0 to 0. So the data sets we use for this, we try simulating data sets to represent typically what we can have in our breeding programs. We have a simple model where we have, like, from each biparental cross, we have one seed, and then for here, we assume like we have like a uniform and random distribution going from parents to progenies. And another case we tried is simulating the second one, but here we use allele frequencies using one of the data from a crossing nursery of our programs, IITA, just getting the whole thing and then using even the number of SNPs. And the next thing, and also we tried to increase the number of progenies, so we have progenies that are related in the scheme. And also the last thing we did was, sorry, I should have referenced, we used the breeding language scheme to bring in a level of, since we're assuming that we have a random distribution and then inheritance here, can we account for linkage? And then in case in a pool where we had before, though cassava is crossing, we'll have a mixture of true crosses and selfing, is there where we can separate the two? So we try simulating a data set that has both true self and then true crosses. We also got data from our breeding programs, NACRI and IITA, their crossing nursery. We had about 379 crosses from NACRI, and then 764 progenies coming from a mixture of 80 parents. And then lastly, we use those methods to check the polycross experiment we had in Umudike in 2015. So the prediction assignment we got one here reporting the three methods, or the two methods, and then for the simulated data set, we were able to account to say, okay, the two parents, there are two recorded parents, how accurate are we in getting two of them? So what we did is to rank the coefficients we got from the two methods, and then the topmost two, we want to like give them the chance that, or assume that they should be the parent of the progeny involved. So for those top two parents, how sure, how do they relate with the written record? So for the simulated results, we had 100% accuracy using the two methods. But when we go to the data from the breeding programs, we had a level of a shift in accuracy is now up to 100%, but then some of them were a bit low. And then for predicting male, so showing like, okay, if they wrote male, how accurate are we in predicting the male that is recorded against predicting the male that is recorded for that progeny. In all, we realized that accuracies were slightly higher in penalized regression than using realized relationship method, even though most of them were still like about the same range. But accuracies in female were mostly better than in male, which ordinarily should be the case because you're harvesting seeds from the female plant. So, well, while we are still asking a lot of questions, this is one of the initial studies. There's still room for development and improve on some other things. But we are looking back on some of the things that can affect or alter your expected outcome. Some of them will range from long receptivity of the stigma, cassava stigma. The normal process, usually you make a cross, you cover for about 48 days, but 48 hours. But now we are seeing that from some experiments that are coming up, especially from CIAT, they are telling us that some of those clones still stay viable or receptive till up to 5 days. So we might have to change. It could be possible that what you crossed was not the one that was taken. And clone mislabeling, that was also affected the experiment we had at Umudike. We gave people a design like row, column, and then somebody is reading plot number in a zigzag method while we gave you from one direction, 1 to 29, and you should come back to start 30 from here, but somebody starts from this side. So those things also, it can also happen when we are cutting cassava, use cuttings, and all that could introduce mislabeling in the field that what you are calling a clone might actually be something else. Bagging conditions, with experience, I realized that it's not like maize where you bag and then throw away. We reuse bags, even sometimes over years. And that is giving us an idea. Maybe we should start asking questions, looking at what is the effect of those bag reusage, whether they are open holes that bees also eventually use to cross a clone that while we cover and think we have covered, they were not really covered. So those are some issues, genotyping error. So we are still trying to understand more about the study. And like I said, this is the initial one. Another thing we ask is, okay, if you want to separate between two crosses and where you have problem, surfing events. So we try to look at the coefficients. So here I'm showing a plot of the topmost number one predicted parent, the coefficient of the topmost parent, and then the second one. So when you plot it, we are seeing a kind of pattern that most of them are within the true crosses are within contribution of 50% from both parents or thereabout. But then for self materials, we are having something higher than that. It's higher than 100 here because we use a percentage of all the coefficients we have. So you might ask, why is it above 100? So with that, also, we try to get the ratio between the coefficient of the first one and the second one. And we realize that if it's about 50% contribution, it should be about 1 or thereabout. But values that are above 3 are ordered. We give them a red sign. Most of them should be self-material. So the same thing goes with the other method. I'm just showing for the lasso method. So in conclusion, we can easily use the abundant marker information to redesign. One, the advantage we have here is that we have access to abundant seeds that we can use to increase our diversity, improve accuracy, and then if we have a good recombination between those traits, that's also giving a good news that we can use the method in current genomic selection scheme. And one other thing is that we can, with this method, be able to have a hold on the true parents of those progenies. And that will give us also, if we are talking about combination in all, from our results, we use 29 parents, and in most of the methods, up to 28 parents, even though we use about 6% of the seeds we generated, showed up as one of the top 2 parents. So what is showing us that we have a good hold of random combination of all the parents we have in our population. So that's good, and then we can be thinking about trying to compare and get combining abilities between those parents, and that can also redefine what we do in cassava. We can start thinking about heterotic patterns in cassava. So in all, I think trying to be time, NIRS can promote phenotyping of known and emerging quality traits. We can use improved phenotyping coupled with abundant genomic information, a good hold in statistical tools to do more in identifying regions that are significant for variation in most of the known traits. And currently we are doing some work to identify some end user preference traits. So we still need to have a good hold of good in phenotyping for all those traits. And then we can use genome-wide predictions to fast-track genetic gain and shorten cassava breeding cycle. And the last thing I showed is that if we use the abundant markers, we have a good result. We are still going to study more and then develop on it. But if we can do that, that gives us a good hold on changing the pattern and breeding method, reshaping and redesigning breeding method in cassava. So I want to just mention, because of our time, some other studies. I was fully involved in implementing GS in NIRS. I have data for calibration for starch and pasting property in cassava. I'll be working on that after now. This is part of the kind of mentoring program. One of the MSc students at the NEXT Gen is trying to compare the other methods that people have developed for TCC, eye check, chromometer, and comparing it with color chart. So I oversaw trying to also help him. He has graduated, and then we have submitted that paper now. And other things we try doing, we have data for that we could be working at the end of the program, trying to standardize that is between the desktop. So CIAT has a desktop NIRS, and we have the portable NIRS, and they have developed cassava calibration base for over 3 years or 4 modern. So we're trying to see how we can standardize between the 2 devices and possibly inherit their database in helping us improve our own calibration model in Africa. And then I was involved in a few studies trying to understand end user preferences and all. So publication, we have some of these are already written and then ready to go. This one was like a collaboration and then trying to put somebody up in NIRS Technology too. So we submitted it just like 2 weeks or 3 weeks ago. And the first study we published in +1 and then involved in general GS and then next gen project. So with this, a whole lot of people, I have the best committee members, and then thanks to our lab members, both Sora's lab and John Luke's lab. I want to thank people like Denise and all the rest of them, Next Gen folks, Kenan is here, everyone. And most importantly, I want to thank the Gates Foundation and maybe Gates himself. I wish I'd seen him one day. I mean, sometimes I think he has the option of using his money for something else, but that's goodwill in trying to invest it in cassava research has seen some of us through in some of what we're talking about. So I want to appreciate him. We have collaborations with a lot of people, ITA, CIAT, NIRS, and ACLI, and all the rest and all. Somebody I want to mention, she was one of the people that encouraged me to apply for. The first time I met her, I was like, well, I think I don't think I know much about GS. She was like, no, go for it, apply, let's see. So I wish she's around to tell her that I went for it and then I'm presenting today. Well, those are some of the old stories and all the rest. I want to thank my family. It was rough. My wife, and I have a little boy that is trying to reshape my life. And it was difficult all the way, especially in 2013. I almost gave up, like one of the shocking moments, lost one of the people that were so close to me, a biological mom. She told me, go, I'll be there to receive you when you finish. So those are emotional moments. I just told myself today I will try and behave myself. So thanks for listening, and then I'll be happy to take some questions. Thanks.
I have time for a few questions before we adjourn with the committee to really drill. I think it was your second chapter or whatever it is. You were talking about you have you're comparing a single-step model and a two-step model, and for some reason the two-step was doing better than the one-step. Now, there's no reason why. I mean, there's no reason why the two should be different, right? Unless either the true values that you're using are different between them, or do you have an explanation for that? I guess I'm really confused as to why they would be different.
So for here, when you are using the single stage model for single traits, is that what you mean?
Yeah, the one, so P is, I got one step. You know what, I got it backwards.
P is one step.
Okay,
so,
I got it backwards, that makes sense.
But that is,
what do P and B stand for?
I just tried to using blob and then raw phenotype somehow. Maybe when I was thinking, maybe I should have used another thing. But I mean, using the single stage, and another thing to add to it, we had reps and all the rest of them because we don't have balanced data. We only use location and set, and we still had like something higher than the two stage. I mean, for one stage should be the ideal thing, but
no, I mean that makes sense. I had gotten them back.
Okay, all right. I don't know if there are people from Geneva.
Any other questions?
Go ahead.
Go for it.
Do you think of Cassava as like taro? I guess that's a Hawaiian root. Do you think of those as similar?
Sorry.
I'm not sure if that's a sensible question.
Taro.
I think it's a Hawaiian root vegetable.
Oh, taro.
Southeast Asian.
Okay.
Southeast Asian also.
Yeah. It's kind of different. This cocoyam, we call it in Nigeria. So
Not that similar.
Yeah, they're all root crops. Oh my God. I thought I have a lot of pictures of roots. I don't have. So this is quite a little bit different, and then taro is always like a little small and then different than this. So cassava is almost like a shrub, and that has another kind of physiology and all that. So they are different.
Different, thank you.
Yeah, but they are all root crops.
In the last chapter, there was a thing where you're predicting which one's the male, which one's the female, and for the empirical data, the female's not 100%, right?
Yeah.
But if somebody recorded which plant they took it off of, and that plant is the correct plant, then that should be 100%, right? So then is that? Can you go to that slide? Is that female value essentially the maximum possible without changing? Without quality, human quality control. And then if that's true, so for example, in your empirical one there, let's say the penalized regression, right? The female is like 80% and the male is 76. So if you think about your ability to actually predict that, it's really a lot better than than you're saying here, right? So I guess, is there a way that you can ask the question of how you should So how you can get those female prediction accuracies to be better from like a human standpoint?
Yeah, I mean, this initial thing, and then when we saw the result, everybody was like, okay, we need to check back and then ask a lot of questions. We had discussion with some of the breeding programs and people, and even with CIAT. That was when Ceballos told us that covering and removing the bag within 2 days doesn't solve the problem. Some things could also happen. But then for female, we actually expected to have like 100% accuracy if everything is all right. So like I said, there are other factors we're not sure of. But we're looking up to see how much we can do. And if possible, going forward, set up more like detailed experiments to see if we can solve some of those things.
But you're right that at the end of the day, it has to be genotype mislabeling or pedigree was reported incorrectly on the table cross.
Right.
And so, we're going to implement a system where when you make a cross, you scan a barcode and it enters the cross into the digital ecosystem.
Okay.
And a lot of things come into play. You are using stem cuttings, and most times these are done by casual workers, and you are having 50, they are cutting every day. One is flying from here to the other point. You can also mix up clones and all that. So a lot of things that we are improving, trying to improve quality control in cassava.
Okay, so sorry.
I had a sort of follow-up on that. How many markers did you need to come up with those prediction accuracies?
So we use the whole markers we're using for GS. I know I know some people have done some studies looking at that you can use up to. I was talking with someone that developed this Solomon package. He said that from his own experience, he can use up to it was as low as 200 if you are sure there is a lot of polymorphism between those traits that you can capture, use as low as 200 or thereabouts. So, but for us, we didn't go into that. Some people have done some studies on that, but we just felt like a simple thing we're using, I still want to run it maybe within my GS pipeline, not going into extra work to genotype or look for specific markers to do that. If I just use a random and then simple, straightforward approach, whether I can do that.
I mean, my observation was more based on the fact that if your accuracies are like that and the cost of genotyping is high, it would behoove you much more to fix the human problems because it's very cost effective and you can improve those accuracies already hugely. Because you're really over-investing in genotyping if those other problems are still or go on.
Not that good. So there is.
Can't fix those.
So Manin is here. He's the quality quality control champion in NextGen. So it's a new initiative. We are asking a lot of questions. We are seeing a lot of things, and we are getting to ask detailed questions. Can we change the way people are doing things? Even in region, can we have a uniform method of reading our fields, collecting data, and all the rest? So those things are in check. We'll try and do that.
I was going to ask basically you guys.
But it's a great, I want to say, I think it's a great thesis the way these hold together. As you pick off problems and give people an idea of how to move forward, you improve things that can be improved, and then the other improvements will come. And I think if you can improve that rate of genetic gain, everybody will be in line to continue to improve what they do. So I just want to say I think you've done a great job of looking at phenotyping, genotyping, crossing, experimental design, all of that in a whole big spectrum. Well done.
Thank you so much.
Doesn't mean we're going to pass.
I think already I have 1 yes from my mind. I have a yes already, so let's see. All right, I think with that, I want to say thank you.
This has been a production of Cornell University. on the web at cornell.edu.